Okay, so in this little PowerPoint, I'm going to go over the stuff you need to know for the last two bits of success criteria. The difference between prokaryotes and eukaryotes in terms of the organisation of DNA, and also a bit about how there's actually DNA in chloroplast and mitochondria. But first things first is you actually need to know the difference between a prokaryote and a eukaryote. Okay, right, first things first, there are three domains of life. There's bacteria, archaea, and eukarya. That means every living thing will fit into one of these three domains. Here we've got a tree showing, a phylogenetic tree showing the evolution of these three branches of life. Bacteria and archaea evolved much earlier than our eukarya, which evolved from archaea. Now, prokaryotes include bacteria and archaea, and they're all unicellular. Eukaryotes include all of the eukarya, which is plants, fungi, and animals, okay? And remember yeast, they often talk about yeast slightly separate, but yeast is a type of fungi as well. So you've either got your unicellular organisms, like bacteria and things, under our prokaryotes, or you've got the eukaryotes, which are unicellular or multicellular, so plants, animals, and fungi. So hopefully from the last task you'll have picked up quite a few differences about our prokaryotes and eukaryotes. Now what you need to know for higher is you need to know about the three domains of life. You need to know that plants and animals are eukaryotes. And hopefully pick this up, eukaryotes have membrane-bound organelles. This means they've got cell structures that are enclosed within a membrane. The nucleus has got a membrane around it. Mitochondria have a membrane around it. Chloroplasts, vacuoles, all these things have got membranes around them. And that's what makes a eukaryote a eukaryote. They contain these cell structures. Now, one thing to note just now is ribosomes do not have a membrane around them. And prokaryotes also contain ribosomes, but prokaryotes don't have any membrane-bound organelles, okay? And a prokaryote, as I've already said, are archaea and bacteria. Prokaryotes are the smallest forms of life that can live independently. And as you see, there's a little pencil there, so usually in class, this might be a little introduction note, so if you wanted to pause this and take a note of it, that's fine, you can always grab it at the end. So what we need to know about in terms of prokaryotes and eukaryotes is how the DNA is organised within these organisms. Right, eukaryotes, whether that's plants and animals, it's organised in the way that you already know about. The DNA is in a nucleus, which is obviously inside the cell, and it's in the form of chromosomes. Okay. Now, new word, or new term anyway, linear chromosomes. Okay, that's just your usual normal chromosomes. We always called them chromosomes. They're now called linear chromosomes. Okay, because they're kind of like two lines. Okay, they're in a line. There's a start and an end to the DNA. Okay, so eukaryotes have DNA organized in linear chromosomes. Now remember, we always said that DNA was tightly coiled into these chromosomes. Now they're actually tightly coiled around bundles of proteins called histones. You need to know about that. You can see that they're little balls of protein that the DNA, it just helps the DNA wind them round, and that's actually how they tightly coil up into the shape of the chromosome. And we've already mentioned this, these chromosomes are stored in the nucleus. Okay. Now this is the main DNA of eukaryotes. This is how basically it's going to dictate how the cell functions. Remember, it's got the coding of life and all that stuff. It's the important DNA. However, it's not the only DNA in eukaryotes. This should also be new information. Okay. So not only is there extra DNA that we've never talked about in Nat5 inside eukaryotes, there's also not linear. It's actually in the form of circular chromosomes. Here we've got our chloroplast, and inside the chloroplast, that's it zoomed in. If we zoom in even more, you've got chloroplast DNA there. And in our mitochondria, if you zoom right in on the mitochondria, it also has circular DNA. Now the reason that they've got their own DNA is because very, very far back in the evolutionary past of eukaryotes, they didn't used to actually have chloroplast or mitochondria. They were actually separate entities. Now what happened is eukaryote cells engulfed the chloroplast and mitochondria and then kept them in themselves to be able to use. So mitochondria, obviously, producing energy from food or breaking down food for energy, and then our chloroplast, obviously, for photosynthesis. Right, as for prokaryotes, you should be familiar with this diagram here. We've got our bacteria cell, and what you also know from National 5 is that bacteria do not have nucleus. Their DNA is just floating free, okay? It's also different from our DNA, eukaryotes, because it's not linear. Not only is it floating free, it's actually a giant tangled circle. There is no start, there is no finish. It is a circular chromosome. And you can see that just here. So if you were to untangle that, it would just be one big loop, okay? And as you also know, bacteria have got other types of DNA in there. Remember, they've got the small circular bits of DNA, which is like extra DNA, known as plasmids. Now, the DNA in plasmids is different from the circular chromosome. It's just extra DNA. The circular chromosome is the main bit of DNA. Okay? And as a side note, yeast cells, which are fungi, so eukaryotes, they sometimes also have plasmids, just to confuse things. But they're the exception. So that was just a quick overview about how DNA is organised differently in eukaryotes and prokaryotes. Okay, so now we have covered this section here. So we should have hopefully covered all of the success criteria for key area 1.